Thank you. The organizer invited me, and also especially Kevin Chen, my good friend. So he asked me to. The title is fixed by the organizer, so it's not my own title. But anyway, I will try to present something regarding the TCM, Chinese Material Medica, the recent progress, and also with the technology advances. This is something like you will see the pictures from ancient times to recent past and modern times. Dramatic changes are being made in TCM. And currently, in the current health system in China, there are two parallel systems, Chinese medicine and Western medicine. Actually, the PRC constitution and law systems really assures the status, which is as same as Western medicine. There's a parallel, two tiers of system. And of course, there's no any other country I can say that to have the traditional medicine system as such a high status and a complete law system. Definitely, not only because of that, and I think unmet medical needs also require TCM, especially for chronic diseases. But definitely we know TCM is a traditional medicine. We need evidence to put on this very traditional, long history medicine system. It's just experience-based. We need evidence for that to turn TCM into an evidence medicine. The general features, I can say there are many other features. This is the one, experience-based. It's not like rational design drugs or others, and also mixture, very complicated system, multi-components. And this feature also assures that, that's, I can say, dominant, that TCM quality control must be different, as Kevin stated. Multi-target, wholly regulating, also some other features. TCM research is really actually approaching in China and made a good progress. So phytochemistry, quality control, pharmacology, toxicology, even system biology recent days is very active in different groups or different universities. I will try to give an overview on the different aspects. Phytochemistry study, I can say, is the most active one, and also many phytochemists in China. From the publication from mainstream journals, Phytochemistry, Journal Natural Products, and Plant Medica, even the organic letter, that's many contributions from Chinese scientists. Many new compounds also published. I can say GMP now at about 25%. Plant Medica really occupies, but Chinese scientific publication, they're about 70%. I think this data is from the editor-in-chief, Dr. Luke Peters. Many definitely. The purpose for this study definitely, we try to follow, I think, this Western approach. Get it pure compound and finally divide that into a new drug. But is that the main way? Or is this, this is definitely in China, we adopt a different approach. It's modernization instead of westernized TCM. So I cannot call it, well, many Chinese doctors will say this is a westernized Chinese medicine. We do not agree. But definitely, I'm open-minded. I think there's a one way, but it's not the major way. This is, you can see many interesting structures published, and they found some very active activities against cancer, virus, and other diseases, but they're still far away to the new drug. But hopefully, there will be a second world-famous pure compound isolated from TCM, just like artemisinin. Artemisinin definitely will be a world-famous one, but whether we can get second, third, or even in a row, in parallel with that. So phytochemical analysis also, I think this is the basis for the quality standard setting. Without complete understanding the phytochemical compositions of the TCM, there's no way that you can control the quality or setting up a good standard. Here we've done some studies, try to compare the several major species of Panax, ginseng, American ginseng, red ginseng. Red ginseng is just the processed ginseng. It's by boiling and there's heat, and nitroginseng is another species. So usually, why I done this research is because there's several species all contain saponins, ginseng saponin, and majority of times the price for these several herbs are very different. American ginseng very expensive, probably the most expensive one. Nitroginseng, about 2 years ago, the price gone up like 700 per kilo, but nowadays it goes down a bit. But still, Chinese ginseng the cheapest. So therefore, there's opportunity that to adulterate these cheap ones into the expensive one. So how to differentiate them? That's a challenge. If after extract or even powder, how to differentiate them, the different species. So that's, I think, is very important. First thing, try to identify those ginseng oxides, and then find out whether or not there are some markers that you can use to differentiate them. But usually it's very difficult. Then you got to adopt a different approach, is that to try to profile them, to profiling the several major markers and even the ratio among the different ginseng oxides. So that's quite successful. We have done this and published papers on that. I think 7 markers can be used for the marker to differentiate the different species, and also try to use the ratio of the different major saponins that you can differentiate whether or not, even after extract, even in the finished product, you can really differentiate whether it's ginseng or nitroginseng or red ginseng. I think this is one of the approach that you can also use to differentiate similar species in other herbs. PK studies. PK studies, before there's no requirement for such. Even nowadays, if you file a new drug for CFDA, there are no such requirement. But if it's the pure compound or it's a fraction, they require this data, this research. But for the combination, there's no such requirement. But nowadays, the Chinese science try to focus on that to clarify some of the PK curves or parameters of the mixture. So we have done some research on the licorice because it's so commonly used. Almost 80% of the formulas have such glycyrrhiza rolensis, gancao in Chinese, to use this herb as, to regulate the whole combination. What we did is try to identify the metabolites of the ADME. So you try to clarify the use of different hyphenated technologies. We can say the state-of-the-art technology. Try to identify all the major metabolites. And finally, definitely, we can identify over 60 metabolites. And also, the second problem will be whether you can draw the PK curves for each original compound and then metabolites. That's very challenging. It's necessary? I don't think so. You draw every single component, but a major one, definitely. You want to clarify those. So here, we try to do some extreme research purposes. So PK curves are 55 components, and you can see you can monitor 64 components, including original and metabolites. And finally, you have all these curves, drawn from these different studies. But this is important. Afterwards, then you need to know that what are the really active components for some specific diseases. So you can see from here the complexity that each component or each type of the component in one herb really goes to the different metabolic pathway, which some of them are really absorbed very quickly, among the 1 hour, within 1 hour. And some of them very slowly. And even some of the saponin glycoside, you can see it goes all 1 day. And after 24 hours, you will not see the peak anymore. But here we shows that the complexity at the one hand. On the other hand, I will see the TCM combinations or the mixtures. Really, the different components in the mixture play the different role at the different time frame. That also shows that why the TCM combination, that they can work quite a long time, even with a very low concentration. That's maybe just an indication or hint, but later you need to really clarify the details on that. What are the major components? But definitely all different components work synergistically, theoretically. Quality control, many scientists, many speakers have talked about this, but as Kevin indicated, I have done this herb research for many years since I came back from the States. Quality control, also many others. I have a basic principle. I said that quality standard, feasible quality standard must come from a basic research or deep research. So without this research, you cannot really draw a very good final quality monograph. I chose this herb because this herb is so popular in China. Many, many products has been derived from this herb, and injections, dancing dripping pill, the tonic pill, and many others. For common heart diseases, you can see the yearly gross sale from this herb, 30 billion RMB, 1 year, simply from this one herb. That's the top herb, even much more than ginseng and other not ginseng herbs. $5 billion, a big industry here. What we have done that is phytochemical analysis first. Try to develop a fingerprint to cover the two different type active components, phenolic acids and danshenones in one chromatograph. Here, you really have an overall kind of monitoring of the different components, including the major ones and also minor ones. With this, and you can really monitor different resources of the herb. You can see the difference. That's really the challenge. That's the problem for the herbal medicine. You need to really to monitor. If you want, you got to recognize that this is natural. But how to use this unnatural, I can say it's a very different quality material and to produce the quality products. That's you need to really monitor the quality of the original herb by choosing some of the different habitat, like those ones are the good ones. If you want to use the danshenones, definitely those are the bad ones. But they're pretty good in phenolic acids. So therefore, for the different purpose, you got to really choose that one. And definitely by processing, by the manufacturing process, you can do a lot of manipulation for the quality. Here, I can show you the state of our technology, RCMS, and even RCMR, and many, many modern technologies that you can use this technology really to identify those major peaks, also minor peaks. So that's several years ago. Nowadays, we can really do this by, even more a modern instrument. So like a water's instrument, a thermal, Angelin, all these new technologies, you can use this to monitor, to identify those minor components within a very short time. And then, secondly, after you make clear the clarification of the chemical components, then you need to develop the assay methods. Probably this will be used either in the pharmacopoeia standard or in the new drug standards. So here we developed the multi-marker assays for the phenolic acids and also danshenones. Combined with the fingerprint and TLC and other type of the technology or the techniques, then you really combine those techniques and finally develop a monograph or the quality monograph. And metabolic analysis of salvia. That's also important. We adopted three parallel technology or research approaches. One is chemical analysis, and then metabolic analysis, and then finally is biological analysis. Biological analysis only helps that to clarify the active ones. So here we show the biological research of this herb, and we adopt the pure compound fraction single herb combination approach. That sometimes pure compound from the major ones will not clarify the formula, even though the single herb. So you really need to combine all this research then to study those type of the components. Here, the single compounds, this fraction, the single herb, and formula with the sanqi. not ginseng. So we have combined all the research, and publication is not important, but we need to know that those are important markers. You need to focus on those to do quality control, to do the assay. Then finally, we draft the quality standard of our salvia and also submit to USP, United States Pharmacopoeia. This is the first TCM quality standard adopted by USP. And now definitely there's some other one. What's in it? Is assay multi-marker, TLC, also very important, and definitely fingerprint is in Chinese Pharmacopoeia, but not in USP. Chinese Pharmacopoeia adopted fingerprint technology, but USP only used as an approach for the study, but were not in the specification. There's some differences. The USP, this is the first one. And then based on this research, since we have done a lot on the salvia and ginseng, then we combine them and develop a new drug now in phase 2 clinical trial. Hopefully, we can get this approved. I mean, it's a long journey, very, very long. I hate to do new drug development. So this approved in 2009. It's still only in phase 2 clinical trial and very costly. Some other TCM quality monograph adopted by USP, salvia, ginseng, gardenia. These several ones are finished evaluation. So this is some new development. USP now have a new move that also adopted herbal medicine in their pharmacopoeias, which is called Herbal Medicine Compendium. Since I'm an expert on the committee, so I know much well on this. So this is some new development. We hopefully that the 99 herbs of TCM will be finally adopted by USP. New drug development from TCM, that is also very, because of the very industry booming in China, so many new drugs has been supported by the industry and also Chinese government and a lot of new products. But in the recent past several years, very rare new products have been approved. The problem is that they increase the bars for the approval, like FDA, like EMA, the same thing in China. But we already have many new drugs in the market. Many are like DL-Shichekang, is already long-lasting, strong marketing. Every year about 6 billion, not 6 billion, it's 0.6 billion RMB every year for many years already. This already shown. Let's always show this as a trial, but it's only one. I hope that many more in the future. So those are two typical products that really have a big market share. Ginseng supplement injection, lyophilized powder, that is 5 billion RMB per year. Really, we can say it's a blockbuster kind of drug, almost $1 billion. Here another one, Danshen. This product developed by our institute, actually in our center by a different professor. This product is booming now every year. This last year is 3.5 billion per year for the sale. This year, almost 5 billion. So that's really, and this guy become rich. It's under my research center. I hopefully, he got very little. He got, if 1%, that's, definitely. But it's only 0.01%, something like that. Scientists always poor. Yes. So we got to really earn our right to get more shares on this. Challenges, I won't talk about it. Many challenges on TCM modernization. Quality, safety, efficacy, I think the most important factors that you got to focus on those. How to really have a quality assured, safety, proof, and efficacy clarified. That's really important. Methodology, mode of action, others, that's, I think compared with this aspect, that's minor. Opportunities in China, I think investment. No money, money talks. No money, nothing. So that's money. Fortunately, that's in China. There's money. The economy situation is much better. And scientists, industry, government, also very enthusiastic. New development research methodologies, innovations, therapeutic requirements, and modern medicine. There are some disadvantages. Therefore, we need traditional medicine. That's what gave the opportunity of TCM. So finally, TCM modernization, what the future? As I said, this is one way, minor. There's the arrow is narrow. Major route, modernization. To keep it is and safe, efficacy, quality, mechanism, clarified. Our focus will be on the quality and modern TCM product development. Now hopefully by doing all of this, then you can have evidence-based TCM. So that's really our goal. Let's find a meeting announcement. Every year organize an annual meeting on TCM natural medicines, which is this year will be 7th year. So hopefully you're interested. Some flyers there. Finally, thank you. That's Shanghai. You can see. It's not now a new building. It's even taller. The tallest here. So there's 3 parallel buildings. But building is not important. We want TCM growing as high as that. Thank you very much.